It's really, want to thank the organizers for inviting me to give a talk. So in CGSI, the organizers in general, we don't put ourselves on the schedule because whenever something happens, we stand in. So since Iran was like last week and I'm today, so to the rest of the faculty, please don't cancel because we're now in trouble. Okay. So I'm going to be talking about discovering causal variance involved in GWAS studies. And there's actually 5 papers that are relevant to this talk, and 2 of them are reviews and pretty accessible. And since I'm going to give you, you'll have the slides, you don't have to write them down now. But one of the reviews is actually on a little bit more than just what I'm talking about today. It's a review that was written to computer scientists and communication of the ACM. Called Discovering Genes Involved in Disease and Mystery of Missing Heritability, and talks a little bit about what I'm talking about today, but also a little bit about mixed models and heritability. And I think it might be of interest to people. Okay, so let's start with the real basics. So what I'm interested in is trying to understand finding SNPs that are involved in disease. And so here's actually kind of a toy example of this problem. And you can see that each line here is an individual, and they have a blood pressure, and you can see that there's most of the positions are black in the genome because they're not different in the population, but there's 2 positions that are variable in the population. And can anyone tell me which of these 2 positions appears to be associated with blood pressure? For here, you guys are good. Yeah, you guys are good, smart. So it's clearly the first position, right? And one way I like to think of it is to draw a plot that looks like this. So on the x-axis, you've got the two values for the variant. So you've got a T and a C. And on the y-axis, you've got the phenotype. And every individual is now a dot somewhere in this plot. And what you're really trying to do when you're doing association study is you're trying to see which hypothesis fits better. One is the null hypothesis that the SNP does not affect the trait. So in this case, the model is just that y equals some population mean. The trait is y, but of course since nothing in this world is perfect, you also have some kind of noise or error, which in genetics is really funny. They've really classified the entire world into 2 things, which is the genes and then the environment. So the E stands for, in genetics, environment, not error. And so this is one model. And then the other model is this model, which is that there's a term corresponding to the genetic effect. And so you've got y equals x, which is the genotype, and beta, which is going to be the actual kind of shift of the phenotype, shift in mean if you have the trait or not. And so when you're doing a GWAS, what you're basically testing is you're saying which model fits the data better. Does this model fit the data better, or does this model? And so if I introduce a little bit more notation, if I just introduce it, so if you think of this in terms of vectors, this is what the model looks like. So you've got y, which is now a phenotype vector of size n, which is n is your population size, and x is a SNP vector, also size n, which is kind of the genotypes in the population, and beta is the contribution of the SNP, and then e is what's called a random effect, which is the contribution of the environment, and the variance of e is going to be, because this is a random vector, it's multivariate, it's going to have a variance covariance matrix, and in this case, it's just going to be the diagonal matrix, where along the diagonal are just the sigma squared e, and off the diagonal is 0. So the reason why we do that is because we really, one of the assumptions we make is that for each individual, we assume that the environments are independent. Okay, so if I kind of add a little bit more complexity, so how does the actual variant in standard models, how does the variant actually affect the trait? Okay, this is kind of, so if I think of now a genotype, or our genotypes are in each of us, you can think of it, you can represent the genotype as a 0, 1, or 2, which is just how many copies of a specific variant you have. For example, if there's a SNP that can be either A or G in the population, you can code the people that have AA as 0, AG as 1, and GG as 2, okay? And you can imagine that some parameter will be the actual contribution of that SNP, which is, so let's call it gamma. So gamma is the contribution of the SNP. So for every copy of the SNP, the phenotype is increased by that amount. So if you go from a 0 to a 1, you've got a shift of that gamma. And what people usually do in genetics is that, because this is kind of difficult to deal with, because this 0, 1, 2, for example, what they do is they normalize all the data in a special way, and I didn't really understand. Like, this is actually a major source of confusion to me when I first saw it. But this is basically what they do. So what people do in genetics is that they transform their model, and essentially what they do is they remap this G's from 0, 1, 2. Instead of 0, 1, 2, which this is 0, 1, 2, they use, wherever it was a 0, it's this value. Wherever it's a 1, it's this value. Wherever it's a 2, it's this value. Okay, and so P is the frequency of that variant in the population. Okay, the reason why they do this is because if you do this, then all of the math works out really, really simply. Okay, essentially what they're doing is they're just normalizing the data, and after this, the math is actually really, really elegant. So. And these are called the normalized genotypes, and it's actually 1-to-1, meaning is that if a genetic variant is associated with a trait in the original 0, 1, 2 model, it'll also be associated in this normalized model, because it's just recoding the values essentially. Okay, but once you do the normalization, then genetics is really simple. Let me show you what I mean. So let's say this is your model here. This is your model. Okay. So if you want to and you have data from n individuals, if you want to estimate the values, for example, if you want to estimate this mu, all you do is you just take the average of all the y's. Okay, that's because the things are normalized. Okay, if you go back to the previous one, you'll see that that's not the case, because you've got these 0, 1, 2, etc. Right? If you want to compute beta, the estimate for your effect size, it's just going to be basically the dot product between the SNP and the phenotype divided by just the average. Basically the dot product and then divided by the number of individuals you have, right? And that's because basically the dot product of the genotypes with themselves is going to be n. And you can actually work that out pretty easily. And then similarly, if you want to compute the statistics, you just apply these other two formulas. And so it's actually really easy to compute the statistics, which is going to be essentially your estimate of the effect size divided by your estimate of this kind of the variance, the environmental variance, times square root of n. So this statistic here is something that you can really easily compute on your data, and once you get it, it'll follow this distribution. Okay, so what is this distribution? There's a normal distribution. Its mean is 1. Okay, sorry, its variance is 1. And then this is its mean. Okay, so what happens if the beta, so beta is the true effect, and beta hat is what you're estimating. What happens if the true effect is 0 to this distribution? So this is going to become like a standard normal, right? Okay, so using this formula, okay, which is just the formulas on this page, you can take real data and you can compute these statistics. And under the null distribution that the beta is equal to 0, it's going to follow the normal distribution. So then you can compute the statistic, and then you can measure to see if the statistic is significant. So if your data follows, if your null distribution follows a normal distribution, your statistics will then fall somewhere in this curve. And if your statistics are really in the tail ends, okay, so this kind of tail end, which is like corresponding to some alpha over 2 on the tail ends, then and if it's kind of beyond that, then the statistic will be significant, will be significant. So for example, here's 3 statistics. Here's 1. Is this statistic significant? No. What about this one? Yeah, and these 2 are, okay, right? So it's hard to see a little bit, right? Do I have to, oh, wait. There's that better? Yeah, okay, you can see that, right? So that's the idea, right? So really simple, and that's what people essentially used for GWAS. And I want to introduce another concept, which is what happens if the beta, the effect size, is non-zero? Then what distribution will the statistics follow? Still normal, right? But the mean won't be 0. Okay, so actually I'll show you a picture in that case. So this is what happens if it's non-zero. So if it's non-zero, what happens is that the mean, the distribution will follow this second curve here, right? And this point right here corresponds to this value, which is called the non-centrality parameter. And then the statistical power of the test, because if you run the test, you get the statistic, and the question is it greater than the threshold? So the probability of it being greater than the threshold is called the statistical power, and then that's going to be this area here. So using this, you can also, using this very simple approach, you can actually compute what the power would be, and people did this. So here's actually a example of the power for different size studies. So this is different minor allele frequencies and different study sizes. This is going from 1,000 to 100,000. So you can see that, for SNPs that are common, you have really high power once you get to these ones, and for SNPs that are and for SNPs that are rare, actually the power goes down. Okay, so just this very basic approach is I think the basics of what people in GWAS did, and this was they did this around, I think all this work around 2005. And just to give you an idea, here's a graph I like to show of just how things took off. Okay, so what is this showing? This is the number of papers that reported a significant association from 2005 to 2013. Obviously it's cumulative, right? It's not like it's cumulative, right? So you can see in 2005, almost nothing reported an association, and by 2013 it was like 2,000. Okay, and so each of these studies basically they did exactly what I described. They collected all the SNPs in a population. They used those they normalized the genotypes. They used those really simple formulas to compute the statistics, and they checked to see if the statistics were above a threshold. Their threshold was set at 5 times 10 to the negative 8th. That's what they decided was the significance threshold. And if they found something like that, then they published a paper, and then the paper kind of got into this graph. That's essentially what it did. And just to give you an idea also of how things have changed over time. So this is the plot that they generated in 2009. This is from the NIH, the NHGRI. They generated this plot, and each of these vertical columns is a human chromosome, and each circle is an association that was discovered using a genome-wide association study, and it was discovered, and you can see that like in 2009, this is what they knew, and so at the time they were extremely happy about this because in 2005 this plot would have been empty, right? So to them this was a lot, right? Just to give you an idea how things changed, this is 2009, this is 2010, this is 2012, this is 2013, and after 2013 they actually stopped making these plots, mostly because if you look at what's going on with the plot. So it's kind of hard to see, but you can see that this is the chromosome, right? And then this is all the results. So there's actually, they didn't stop making the plot because they want to make the plot, but they actually didn't have enough space on the chromosome to list all the marks, right? So so that was a great success, great success. But then, so what we actually tried to look at was a problem kind of follow on to this, which is that, okay, so they found all these associations, right? And so they did this GWASs, they found these associations. So the question is, can we detect what the actual causal SNP is? And I'll kind of talk a little bit more about that. But what do I mean by that? So if we actually, let's say this is for example, consider this plot here, right? This is called a Manhattan plot. These are the chromosome numbers, 1 through, 1 through X. And the height of each of these points is a SNP. The height of each of these points corresponds to the negative log p-value. So anything that's above this line is considered significant. And so you can see that there's a really sharp peak here, right? So this looks like a success of GWAS, right? Because you say, and then you can say, well, what is the actual gene, et cetera, right? But if you zoom into that plot, this is what it looks like, okay? So it's not that there's just one SNP that's significant, but there's many, many SNPs that are significant. And they all look about the same. So it's very hard to say which SNP is it, okay? Or is it more than one SNP? This is the question, right? And this is actually, and oftentimes these regions where you have a lot of significant SNPs, they stretch across many genes. So actually, in fact, some of the early exciting reports of this gene causes this were later found out to be the wrong gene. It was like another gene, right? And so let me explain why you have this phenomenon, okay? Why do you have this phenomenon? So again, it's not like, let's say one of these SNPs, so here's the question. One of these SNPs is causing is actually affecting the trait. It's actually that there is a SNP in the region that if you have it, the mean of your trait will be higher than if you don't have it. So there actually is a SNP that's causing it. So the question is, why, when you see that, do you have many, many SNPs in the region? Okay, so if I kind of go back, here's actually a single SNP, let's say it has a certain beta. So this will be the statistic. The statistic this is a kind of review, this statistic will follow this distribution. So it'll have some non-centrality parameter with variance of 1. And of course, because we're assuming that the SNP has an effect. So now, let's add in a second SNP in the region. And let's say the correlation between these two SNPs is R, okay? And what I mean by the correlation, just take the two vectors of the genotype vectors, or basically the value of the SNP in the individuals, compute the correlation between those two vectors, and that's what I mean by R, okay? So very simple. So if you have two SNPs that are correlated, and so it turns out that the SNPs are correlated, then the covariance between the statistics themselves is also that same correlation. So if you've got the situation where you have two SNPs, one of them, let's say SNP A is the causal SNP, and if you've got this vector of SNP A and SNP B, it'll follow this multivariate normal distribution where this will be the non-centrality parameter corresponding to the causal SNP, but its neighbor will also have this non-centrality parameter, which is the same effect size at the SNP times the correlation. Okay, so first of all, what happens if the correlation is 0? So yeah, so then this will be a 0, right? Okay, but if it's not 0, then they'll have some value there. And actually, the covariance is also has a really nice form, which is just the covariance is exactly that value. And if we then think about, okay, so that's the basics. So then if we then think about, kind of in a locus, let's say you have M SNPs in a locus, we can model what we call the causal status, which is just which subset of the SNPs have an effect. And in this case, let's just say that we have M possible SNPs. There's going to be 2 to the M possible combinations of which ones are causal ones. And So if they have an effect, we can say that that entry is 1, and if they don't have an effect, is 0. So for example, here's an example. Here's a 5 SNP. Let's say your region has 5 SNPs. What does this mean? This means if 0, 0, 1, 0, 0 means that the third SNP is the causal one and the other ones are not causal. Okay, so if we assume this, if this is, let's say, that what's actually happening, if we kind of extend that pairwise thing to the entire locus, the statistics in the entire locus, so overall like M SNPs, will follow the multivariate normal distribution, which is the effect of the causal SNP times, this is the correlation matrix between the SNPs, times the causal statuses, and this will be the covariance. Okay, so this is just I know I'm kind of building up to this, but the second review on the in there really just goes through all the derivation. But what is this saying? This is basically saying that, you know, what this distribution of the SNPs in a region will depend on 2 things. One is which subset of them are causal, right? Which one, and how are they correlated with each other? So if they're highly correlated, those ones will kind of move together, and the ones that are not correlated, those will be separate. Okay, and so this is really what explains the following phenomenon. So because you have relatively high correlation in each locus, then you know, even if you have one causal SNP in the region, it'll kind of make a lot of SNPs that are causal. Okay, so this is okay. So I just explained to you why this phenomenon. I just explained to you why this is hard, and I've also kind of hinted why this is not a solvable problem. And so that's why we kind of went into trying to work on this problem. Okay, and this problem is called fine mapping. Okay, so the fine mapping is you know, we have a GWAS, we have associated statistics, and we want to identify the causal variance, and it's called fine mapping. And so the questions we're really interested in understanding are which variants are causal, and that's fine mapping at the variant level. Which genes are causal? How many variants are there in a locus? That's also called allelic heterogeneity. And if the same variant is causal in 2 studies. And kind of we'll go through these. Okay, so but before that, let's try to get some intuitions. Okay, so I explained to you why fine mapping is hard, but sometimes it's not hard. So let me give you an example. So if you look at this, this is, let's say, the result of your study. And down here, this part right here, this is actually this is a representation of the LD matrix. Okay, this is the correlation matrix, but tilted on its side. And so like the darkness of the point corresponds to the dark, you know, like how correlated something is. So for example, this is a relatively light color, and that means that this SNP with this SNP is not correlated. This guy's a dark color, that means this SNP and this SNP are correlated. Okay, so if I gave you this result, right, of the association study, and I asked you which one is the causal SNP, which one would you say? 33 or 35? It depends on your eyesight. I think it's, yeah, 32 it looks like. 33, okay? Okay, so yeah, that's very good, okay? You guys are right. Okay, so, and I know that because it's simulated data, so I know it's correct, okay? Okay, but in practice, so in this one, how about this one? Okay, so which one is the causal SNP? You know, I'll give ice cream to anybody who gets it right, yeah. Okay, so actually, there's no way to tell, right? And actually, what we just attempted is the most common approach to doing fine mapping, okay? Which is, I kid you not, looking at this plot, and then trying to say which is the causal. It's true, right? That's usually what people do, okay? And so what is fine mapping? Okay, let's kind of formalize the problem a little bit. And so given the marginal statistics, we want to predict what the actual causal SNPs are. And the challenges are is linkage disequilibrium, that's That's what people in genetics call correlation for historical reasons, yeah. And there also could be multiple causal snips in the locus. We'll talk about that. And the kind of existing methods we'll talk about are like, we'll talk about them in a second. Okay, so what do people usually do? Here's actually an example of what people usually do. They just say the top K method. They'll say, okay, I have an GWAS. I assume that my causal SNP will be in the top SNPs. There's actually a reason that the causal SNPs in general, especially if there's only one causal SNP, is most likely to be your top SNP. Your top SNP is the highest chance of being causal. But they'll just say, so they'll give it some wind. They'll say, okay, I'll pick 5, and I'll hope that the causal SNP is in the top 5. Okay, that's one way. So the problem with this approach is that it really ignores the LD, and it also it doesn't. You can't really pick the value of K. And then the other thing is that if there's more than one causal SNP, oftentimes the highest SNP doesn't have to be if there's like in this case. And actually, I'll kind of give you a hint here. So this example, these 2 SNPs here are the causal SNP, okay? And they actually induce this SNP being the highest SNP. But it's actually, that's not a causal SNP, okay? So that's why top SNP. So does anyone know how okay, so another approach is called the conditional method. Have people heard of conditional method? Okay. So what people do, and they basically take the most significant SNP, and they pick it, and then what they do is they condition on it and recompute the p-values for the remaining SNPs. So that's what happens here. And then they take the most conditional next SNP, and then they pick that and they kind of repeat until at some point when they stop. Okay? I mean, okay, but people really believe that this works, okay, in the field. And so let me give you some intuition on why that doesn't work. Okay, so what's the problem with the conditional method is that you're making an assumption that the top SNP is the causal SNP because you've picked it, right? So how often is the top SNP the causal SNP? And actually, here's an example showing that. So these are 2 different regions, okay? This is a region, the same one as previously, which has a lot of LD, a lot of correlation. And this is a region that doesn't have a lot of correlation. And so what we did was 1,000 times, we picked a SNP at random in the region, implanted an association, computed all the statistics, picked the top, okay? And actually, we didn't pick the top. We actually ranked them all by their significance, and we drew a histogram. Where does the causal SNP fall in that ranking? Okay, so the idea is that the causal SNP should be the most significant. If it's always the most significant, then that's great, because then the top SNP is the most significant. And what we see is that it's not the case. So in this scenario, actually, it looks pretty good, right? Almost all the time, the top SNP is significant, right? But in this scenario, you have a very large number of times where the causal SNP is actually not significant at all. Okay? Yeah. Yeah, no, it's real SNPs. No, it's real SNPs, yeah. And also, you have to remember is that, as we sequence better and more SNPs, you have things are a lot denser, so you have, more and more LD, right? Right, so yeah. Well, no, okay, so they can, no, but okay, so I think I don't understand exactly what you're saying, but I think you're trying to fix a method that's like irredeemable. Yeah, that's my yeah. Oh, you're saying that pick the. Yeah, I mean they could I guess. I mean the problem is that they believe. I mean there's just because you pick the top one. The rank. My point with this slide is that the ranking of the SNPs doesn't really tell you that much about which one is causal, right? So it's there's not that much information there. Okay, so this isn't going to work. This isn't going to work. And actually, we can see that looking at this plot, right? The problem is hard, right? Because what does this say? This really says that there's really ambiguity in the data, right? It's kind of by chance. Sometimes, the top SNP, the causal SNP is the top SNP, but sometimes it's not, right? And that just has to do with the fact that things are correlated, and so our idea is something we're actually going to try to solve an easier problem, which is instead of trying to find what is the causal SNP, we're instead going to try to come up with a set of SNPs that we think the causal SNPs will be inside that set. So this is easier, right? Because if we wanted, this gives us some ability to make some errors. Okay, we call this the row causal set, and so it's the set which contains all the causal SNPs with at least probability row. So in practice, we want to have this to be like 95%. So and we don't guarantee that all the SNPs in our set are rare, meaning that we can pick things that are not causal, but not the opposite. We don't want to leave anything out of the set that could be causal. So a really trivial solution is to pick all the SNPs as the causal set, because that'll really satisfy this criterion. But that's obviously not what we want. So what we want is we want to pick the smallest causal set. We want to pick the smallest set of SNPs that satisfies this condition. Okay, so let me show you, go back to that previous example. So this is when and I'm going to show how the method works and stuff like that, but when we apply the method, this is what we get. It picks these 7 SNPs, and you can see that the 7 SNPs contain these 2 SNPs that are causal, right? But then there's going to be another 5 SNPs that are not causal, okay? And so you can see that, like, looking at this, right, there's no way that I would have picked this just by looking at the data, right? For example, why did it decide to ignore all of these ones, right? Even though they're higher than these ones, right? But somehow it knew. And it wasn't like I kept on looking for an example where it worked. This is actually right. Okay, but it's because there's some information in the combination of the statistics and the correlation that lets it differentiate between these SNPs. Okay, so let me explain how the method works. So here's the idea. Yeah, go ahead. No. This is all single causal SNP. It's just because imagine if their correlation is 0.999999, for example, between 2 SNPs, right? So then 50% of the time, one will be higher than the other, right? That makes sense? Yeah. So how does this work? So here's the idea. So given in a region, you have 2 to the m, and every SNP can either be causal or not. So you have 2 to the m possible kind of scenarios describing what's actually going on, right? Causal statuses, right? And so you can imagine this as a vector. So I have 2 to the m possible them. And so now let's say I'm going to pick a set of k SNPs, which I'm saying that the causal SNP has to be in that set. All the causal have to be in that set. So which of the causal statuses are consistent with that set, right? Well, if all the SNPs, all the essentially ones of my 2 to the m have to be in that set, right? Then that means that everything outside in the m minus k SNPs, those must all be zeros. So that means that the number of configurations, causal set statuses, that are consistent with my set is going to be 2 to the k. So I have 2 to the m, that's a full space, and I've got a set of k SNPs, and now I've got 2 to the k possible subset of those 2 to the m vectors that are consistent with that, right? And so the way that our method works is basically the following: is that given a region with m SNPs, given a region with k SNPs, we kind of compute a posterior probability that the set contains the causal SNPs, and I put everything in quotes because there's a lot of kind of caveats and assumptions here, but they're not really that important. Unless you're like Bayesian and you really care about these things, but I think they're not that important. And essentially, this posterior probability is essentially the sum of the likelihoods that are of the terms that are consistent with the 2 to the k to the set, divided by the sum of the likelihoods of the 2 to the m. So the intuition is that if this is high, really close to 1, what does that tell you? It tells you that the majority of the likelihood is dominated by these configurations, right? So it's probably one of them, okay? And what those are really saying is that your causal SNPs are inside that set K. And you can imagine that you can just compute this for every possible set of K. And then if you find one that satisfies this, then you have pretty good confidence that these are the right SNPs. Okay. So I'm going to skip a little bit. Okay, so and then if I look at the existing methods, and then I just want to mention that this kind of using a set is not really our idea. There's this concept of credible sets, which they also proposed previously, and a lot of people now kind of use this idea. And just to give you a sense, so how does it work? So if I look at simulated data. If you look at, just what's the recall rate? The recall rate is how often is inside your set do you find the association, and you can see that CM is a conditional method. The conditional method really falls apart, particularly when you have more than one thing. But like our method works well, and then this. Actually, can I go ahead? Okay, so okay, I'm gonna skip result. This is CGSI. Who cares about results? Okay, so let me talk about a few interesting things that you can do with this method. Okay, so there's another question, which is you want to combine. You have GWAS data, and then you have expression QTL data, and you want to combine this. And this is the reason why people want to combine this is something called co-localization. And so what's the idea is that most GWAS they find in non-coding regions of the genome, and so the hypothesis is that these variants affect the phenotype through their effect on gene expression. So the idea is that the SNP affects expression, some regulatory network, and then that has some downstream consequences on the phenotype. And so if a GWAS risk loci is the causal loci, then it should also affect expression for a specific gene, at least in some tissue at some point in development. Okay, so because if the SNP doesn't affect expression in any way, then this hypothesis can't be true, right? So the idea is that the SNP should be affecting expression. So now if we have, and so co-localization is a fancy name for identifying the scenario where the same SNP affects two different traits. So in this case, a GWAS and an EQTL. So let me give you an example. So here is a GWAS and an EQTL, and here you have one scenario where the peak SNP is the same for the two, and there's a lot of evidence that this is the actual causal SNP because there's nothing really around it, right? So that really suggests that it's the same SNP that affects these two, okay? Here is actually another example. You have this, actually the peak SNP is also the same, but are you sure that it's the same SNP in these two, in these two studies? No, right? Because it could be, it could be the one, but for example, it could be this one, but this just happens to be the big SNP, right? So in this case, it's kind of an ambiguous case, right? So that's the intuition, right? And okay, but if this method was working, you can do the following. So here you have a GWAS loci, and then you have 4 genes that are around that GWAS loci, and you have 3 tissues. If you can compute the co-localization probability for each gene in each tissue, and you saw something like this, what would that tell you? You saw some very high values. What would that tell you? How does that? What's the mechanism of that SNP? So, for example, does the SNP affect gene 1 in the liver? Is the mechanism of the SNP? No, right? So you would say that this gene, this GWAS SNP, appears to affect through gene 4 in the liver and the blood, right? That's the idea. And so how do we do that? And so it actually turns out that to compute this probability is really simple. It's just, everything decomposes to that if the variant is causal in each study, it actually decomposes to the product that are being causal in each one. And we can compute this. And actually, I'm going to skip this, but basically what we were able to take GWAS and combine the Gtex data and identify the SNPs to split all the GWAS loci into 3 categories. One, they're variants that co-localize. One that are variants that don't co-localize with any tissue, and ones that are ambiguous. Okay, so can someone guess what percentage of variants do you think co-localized? That we actually found so the GWAS variants that were also in EQTL. No. Close, but no, yeah. Huh? 5%? Okay, what percentage of people do you think that did not co-localize? Huh? 95? No, okay. So I'll tell you, okay, so what we found was that 10% co-localized, 10% do not co-localize, and about 80% were ambiguous. So the ambiguous we don't know, but people were actually shocked that they don't co-localize because they collected the Gtex data and all these EQTLs, they just assuming that they co-localize, right? So that was that. And so when we were worried, we were saying that, how can they not co-localize? That makes no sense. What is that saying? That's saying that the GWAS SNPs are actually affecting traits using some new mechanism, right? That doesn't affect expression. That's really a weird. That's a weird result. That's not what people thought. So then we started to try to answer that, and what we looked for is the following. We looked for what's called allelic heterogeneity. So what is allelic heterogeneity? That's just a very fancy name for the following phenomenon, is that in a locus where you have a gene, for example, what you have is you have more than one causal SNP, okay? So if you see this region, how do you know that there's more than one causal SNP in the region? Right, because there looks like there's 2 peaks, right? And they're not correlated with each other, right? So that really suggests that there's more than one. But then what about but what do people usually do? What people do usually is that they actually do the conditional method. What they do is take the peak SNP, they condition on it, find the residuals, and they keep on doing this until it's significant. So this is what people most of the time report. And so what we did is we extended the fine mapping approach to this problem, and we did it the following. So so we have the causal statuses as we have last time, and what we did is given a region with M SNPs, we can compute the probability of having exactly K causal SNPs as the following. We can look at all configurations that have exactly K1s and sum all of them, and then divide that by all the likelihoods. And we can do this for every K. And so what this will give us is a histogram of what's the probability of having each number of causal SNPs. And then if K is greater than 1, if there's a lot of probability for greater than 1, then we can say that that region has more than one causal SNP. So this is actually kind of an interesting approach because previously people had to identify, if they wanted to identify that there's 2 causal SNPs, they had to actually say SNP, this SNP and this SNP are causal. Here we don't actually know exactly what the causal SNPs are. We're kind of summing over all the possibilities of more than one SNP. And when we compare to the conditional method, obviously it works really well because the conditional method is really bad. When we applied it to the Gtex data, so this is actually an example. So here's actually one gene in the Gtex data, and then this is the caviar set has one SNP, and this is our histogram. What does it look like? So in this one, you see that there's a very high probability of only one SNP. So in this case, we kind of assume that there's only one SNP here. But here's actually another example. This is an example, so it's kind of a broader peak. And our method really says that it's really likely 2 or 3 SNPs. And what does that mean? That means that there really is no one SNP that explains the observed signal. Okay? And then what we did is we applied this to all the Gtex data. And what I'm showing here is this here is the number of genes that have allelic heterogeneity, and this is a function of the number of individuals that they have that tissue for. And you can see that it's highly correlated. Okay? What did that tell us when we saw that? Okay, so this is the number of SNPs, number of genes that have more than one SNP, okay? And then this is the number of individuals that we have tissues where we met. So each of these points is a different sample, okay? That sample has a certain number of individuals, right? And I'm plotting here how many genes we found with allelic heterogeneity. So what does that tell us? What does this plot tell us? Exactly. So really what this told us is that it's probably everywhere, right? And it's just we're only limited in seeing it based on how much samples we have. Okay? And then also we actually looked at 3 different data sets of blood EQTLs, and we found that there's a lot of overlap in where we find more than one SNP. So it's not like an artifact of data. And then finally, even more surprisingly, we looked at GWAS data. And so we looked at schizophrenia and HDL cholesterol. And so out of the 108, remember for a while there was like this famous 108 loci in schizophrenia, right? So 25 of them had allelic heterogeneity and 13, so basically a third. But really, as the sample sizes get larger and larger, you have allelic heterogeneity and everywhere. So what we found is that essentially almost everywhere we looked, there's more than one causal SNP in virtually every locus, and it's just a limit. It's just a number of time, just sample size. Actually, it was really funny when we had the experience when we submitted this paper for review. We had one reviewer that was like, yeah, he liked it, but of course, they're so negative. So even though, he said all these nasty things, but it was overall positive. Then we had one reviewer that said, oh, you have to reject this paper because everyone knows this, right? And then we had another reviewer that said, you have to reject this paper because this is definitely not true. So, okay. So, so this is kind of what we saw, and if I have to talk again, I'll talk about one more topic, but I'm not going to, unless someone cancels. And so I just really want to acknowledge Farhad Hormozdiari. He's actually the brother of Faridun. He was like the real driver on a lot of this work, but it was a collaboration with many other people, including Bogdan's group as well. Okay. Thank you. Yeah. Okay, so okay. I want to answer your first question. So they could be. I mean, we're generally looking in like a gene region, right? So sometimes they're really close to each other, sometimes they're not. If they're really correlated with each other, like extremely, we can't really differentiate between the two of them. But oftentimes, everything is slightly correlated. So I think that methods that require 2 really independent peaks, they almost found nothing. Okay, what do you mean like? Could we change co-localization by window? What do you mean by that? Oh, I see. No. So yeah. So the difficulty is that when you have more than one causal SNP. Actually, I'll give you kind of a thought experiment why co-localization is really hard. Let's say you have 3 SNPs. Let's say, no, actually even the following situation. you have 10 SNPs that are perfectly correlated, and they're completely not correlated with anything else, okay? And then you have 2 studies where they're significant in both of the studies, right? So then, is it the same SNP or not, right? So I think your answer to that really depends on if in life you're an optimistic or a pessimistic person, okay? If you're an optimistic person, you're gonna say, oh, well, in the whole genome, there's so many possible SNPs, right? What's the chance that, it happens to be one of these 10 in both of the studies? Then it must be the same SNP, okay? So that's one situation. The other one is saying that, well, look, there's only a 10% chance that it's the same SNP, right? So I think that's kind of a lot of the difficulty in co-localization. Yeah. Oh, I see. So in our method, we're really assuming that it's things are either causal or not, right? And we're assuming, and actually in practice, because there's a prior that most SNPs aren't causal, you're really talking about like 5, 6. 5 is like the highest. That's all the other terms don't really contribute much to the likelihood, right? There's another model that people have been, this kind of Fisher polygenic model, where every SNP contributes to the traits, right? And so that actually makes a lot of sense when people analyze kind of globally the entire trait, but in a specific locus, it doesn't appear to fit the model. So yeah. So I think also people reconciling these kind of heritability models with these kind of local, models, that's like something that I think people are working on in the field. Yeah. Yeah, I mean, we do, we do, so 2 things. We first of all only allow up to 6 causal SNPs, right? And we actually computed all of them. But then if you take, well, no. Well, I mean, also the other thing is that another trick you can do is, let's say you have 1,000 SNPs. If you compute anything that's not in your top 50, for example, or top 100 is not really going to likely to be there. But then, there's actually an entire cottage industry of new methods using importance sampling and all sorts of things to speed it up. So there's a lot of work. A fine map is a method out of Benner et al. And then there's I think Bogdan had a method. Is he here? I don't know if he's here, but he had a method, I think, on importance sampling. So there's a lot of approaches to try to speed it up. We actually found that in practice, if there's so much correlation and things like that, the results aren't, you're not going to, if your output is 112 SNPs, then that doesn't really help you in general, right? So in the cases where it's useful is when you get down to 10 SNPs or so, and in those cases, it's actually usually not that hard to compute. Okay, yeah? Yeah. I mean, so yeah, so the issue I think would be if you condition on what the actual causal SNPs are, right? Then the signal should go away, except you'll still have noise. I think because you might be conditioning on more things, right? It's not. I don't have to think about it. But that's yeah. You have friends that do conditioning, right? You're trying to speak up for them, right? Yeah. Okay, that's it. Yeah. Okay, pizza at 6, right? So.